On lecture 3, we'll talk about the genetics of microorganisms. Means we are going to talk about DNA molecule. First, we stop on DNA structure. Then we continue with a function of DNA molecule. Means we'll talk about replication and gene expression. And at the end of this lecture, we'll talk about mutations. We start with the structure of DNA, and today we are going to talk about structure of double-stranded DNA. Any double-stranded DNA can be described by three main characteristics. Number one, all double-stranded DNAs are double-stranded, means they're composed of two strands. Number two, all double-stranded DNAs are complementary. We will talk about it. And the last one, all double-stranded DNAs are anti or anti-parallel. So as I said, all double-stranded DNAs are double-stranded. Why? Because they are made of two strands. Now let's talk about what each strand is made of. Each strand of double-stranded DNA is made of nucleotides hooked together. Each nucleotide consists of three parts: deoxyribose, which is sugar, phosphate group, and nucleoside bases, or we just call them bases. There are four bases in DNA molecule. Those are A, adenine, G, guanine, C, cytosine, and T, thymine. On the left side of this slide, you see formula of deoxyribose, sugar. As you see, deoxyribose is not symmetrical molecule. Also, you have to know that deoxyribose molecule has 5 carbons in its structure, and each carbon has its own name. For example, this one over here is called 1' carbon. This one over here is 2'. Over here will be 3' carbon. This one is 4' carbon, and the last one, this one is 5' carbon. And phosphate groups connect deoxyribose or sugar molecules within strand. But you have to remember, because deoxyribose molecule is not symmetrical, phosphate groups always connect 3' carbon of one sugar to 5' carbon of the next sugar. Let me repeat it. Phosphate groups always connect 3' carbon of one sugar to 5' carbon of the next sugar. 3' carbon of one sugar to 5' carbon of the next sugar. DNA strands also are connected to each other within DNA molecule. Strands linked together by hydrogen bonds. Hydrogen bonds only can be formed between complementary nucleoside bases. And please remember, adenine is always complementary to thymine, and guanine is only complementary to cytosine. That means hydrogen bonds only can be formed between those bases A, T, G, C. This is what we call complementary part of double-stranded DNA. Let's repeat it once again. Hydrogen bonds only formed between complementary bases AT and GC. So let's look at this slide. If one strand of double-stranded DNA will look like this, CT, AG, CAT. How would the second strand then look like? That's right, it would be GA, TC, GTA. And the last one, phosphate groups connect deoxyribose molecules within each strand in certain specific way because sugar molecules are not symmetrical. So that's why phosphate groups always connect 3' carbon of one deoxyribose molecule to 5' carbon of the next deoxyribose molecule within the strand. Now we know why double-stranded DNA is called double-stranded, because it's made of two strands. We know why we call it complementary, because hydrogen bonds only can be formed between complementary bases. And the last characteristic, we call double-stranded DNA anti or anti-parallel. Why? Let's look at this picture. Because deoxyribose sugar molecule is not symmetrical. In one strand, it will go in one direction. You see oxygen molecules of sugars of this first strand go up. If you look at deoxyribose molecules at the second strand, you see oxygen molecules go down in this strand. That's why if this strand has 3' carbon on this end, we call this end 3' end. And then on the opposite end of the same strand, it's going to have phosphate group, and that's why we call this end 5' end. But the next strand will have opposite ends. It's going to have 3' end on this side because it will have 3' carbon on this end, and 5' end on opposite side of the same strand because it will have phosphate group at this end. Alright, now we know a structure of DNA molecule. We can continue with reactions of DNA. In order to fulfill its biological role, DNA molecule enters two kinds of reactions: replication and gene expression. The replication is the process by which a DNA molecule is precisely duplicated. Gene expression, the process by which the information stored in DNA molecule is used to tell cell what to do. Today I will talk about how bacteria make proteins. There are two steps in gene expression. Those are transcription and translation. On the picture of this slide, you see once again two reactions of DNA molecule: replication and gene expression. Cells go through replication when they divide and produce two new cells. That's why the end product of replication will be two copies of DNA molecule. And gene expression consists of two steps, transcription and translation. And as I said today, we will talk about how bacterial cells make protein. We start with replication. The first step of replication is called initiation. It starts when in small region of bacterial chromosome bubble is formed. What will form what we call bubble? Bubble is formed by specific enzymes. You don't have to memorize their names. Within that bubble, two strands of double-stranded DNA molecule will be separated. When two strands of double-stranded DNA are separated, bases will be exposed, and 2 points of separation are called replication forks. Let's look at this picture on this slide. On this picture you see what we call bubble. You see within this bubble two strands of double-stranded DNA molecule are separated, bases are exposed, and points of separation are called replication forks. So we have one fork here and one replication fork over here. And then two new strands of new DNA molecule will be formed within bubble. This step of replication is called elongation. When two new strands get formed within bubble, the old strands will be used as a template, and new strands formed within that bubble will be complementary copies to old strands. So what is the name of this substrate that will make those complementary copies of DNA molecule? It is an enzyme, and the name of that enzyme is DNA polymerase 3. So it is DNA polymerase 3 adds new complementary nucleus side bases and forms complementary growing strand. Please remember, DNA polymerase 3 only can build up new complementary strand moving from 5' to 3' direction. So if you look at this picture here on this slide, this is our growing strand. Remember I told you DNA polymerase 3 only can move from 5' to 3' direction when it builds up new growing strand. That means for this new growing strand on this picture is going to be 5' end on this side and 3' end on this side. And because it is complementary copy of old strand, for old strand is going to be 3' end on this side. 5' end on this side and 3' end on another side. Let me repeat it. Because DNA polymerase 3 can move only from 5' to 3' direction, it means for this new growing strand is going to be 5' end here and 3' end here. For old strand it's going to be 5' end on this side and 3' end on this side of the strand, because growing strand it is complementary copy of the old strand. Because bacterial chromosomes are circular chromosomes, after bubble is formed, replication forks will move in two opposite directions. And then elongation, the step of replication, continues, and opposite directions until replication forks meet at certain point. We call this point termination point. It means two copies of DNA are made, and then two strands get separated, and each new chromosome molecule will be made of one new strand and one old strand. Alright, let's talk about DNA polymerase 3 once again. Remember, it is an enzyme that makes complementary copy of DNA molecule. So each DNA has one end, we call it 5' end, with sugar phosphate on that end. If that strand has 5' end on one side, on the opposite side it will going to have what we call 3' end with hydroxyl or OH group on opposite side. So DNA polymerase 3 can only add new nucleotides and build up new complementary strand when it moves from 5' to 3' direction. Let's look at this picture. You see double-stranded DNA molecule on this picture. The first strand has 3' end over here, and you see there's a hydroxyl group on this end. So an opposite end of this strand, of course, is going to have 5' end with phosphate group. And because double-stranded DNA is anti-parallel, the next strand will have 3' end on this side with hydroxyl group and 5' end with phosphate group on the opposite side. So DNA polymerase 3 only can add new nucleotides when it builds up new strand from 3' end side of the strand. Why? Because it can remove hydroxyl group and then add new nucleotide from this side. DNA polymerase 3 cannot dismiss phosphate group at 5' end because phosphate group is too heavy for DNA polymerase 3. That's why we say that new growing strand always will grow from 5' to 3' direction because DNA polymerase 3 builds up new strand moving from 5' to 3' direction. Let's look at this picture of replication. It has a little bit more details. We know two strands of DNA molecule run in opposite directions. They are anti-parallel. We also know that DNA polymerase 3 can move only from 5' to 3' direction. It means only one of two strands at the replication fork can be extended continuously in the same direction that replication fork is moving. This strand is called the leading strand. The leading strand. As you see on this picture, because leading strand has 5' end on this side and 3' end is always going to be on this side, it's going to be not a problem for DNA polymerase 3 to add new complementary nucleotides and build up this new leading strand, moving to the same direction as a replication fork. But the other strand, called lagging strand, must be replicated in the opposite direction to the fork movement. Why? Because lagging strand always will have 3' end on this side and 5' end on this side. And since DNA polymerase 3 can move only from 5' to 3' direction, it means replication of lagging strand always will go in opposite direction from replication fork. How is this possible? It's done by synthesizing short pieces of DNA. We call them Okazaki fragments. So basically, DNA polymerase 3 starts, let's say, from this point of DNA molecule, and then while moving from 5' to 3' direction, forms this first Okazaki fragment. Then it moves backwards, starts somewhere over here, and again moves from 5' to 3' direction, makes this Okazaki fragment, and then again goes backwards, starts somewhere over here, and makes this next Okazaki fragment. And then after all Okazaki fragments are made, one more enzyme called DNAligase, it is on the bottom of this slide. So DNAligase will connect all Okazaki fragments into one solid strand.